This video will guide you through the swab method for identifying individual deer from fecal samples from the field into the lab. By following the steps in this video, you will learn how to properly collect successful samples. Make sure you have all the supplies needed prior to searching for deer pellets. You will need 2 milliliter snap top plastic tubes pre-filled with Longmire buffer, a simple salt solution that can be made in most basic molecular labs. These tubes should be filled before heading into the field and kept in a box for compact storage. You will also need sterilized swabs and packets, etcher for labeling sample tubes, field notebook or data sheets, pen for recording data, and a GPS device for recording coordinates. You will search for deer pellets while walking a transect or in a designated area based on your particular established protocol. Ideally, fresh samples are better, but you can get high-quality data even from older samples. We recommend collecting any pile with intact pellets that are not highly degraded. Once you have found a deer fecal pile, it's time to collect. With the swab collection method, we do not collect the pellets themselves, but rather the layer of deer epithelial cells on the surface of the pellets. First, remove a swab from a sterile packet and take out one of the pre-filled 2 milliliter tubes. Dip one end of the swab in the buffer, wetting the swab. Rub the swab on the outside of at least 6 pellets in the pile if possible, dipping into the buffer occasionally to re-wet the swab. Apply enough pressure to the pellets to ensure that the outside layer of the pellets is transferred onto the swab tip. Since you want to maximize the transfer of cells, you should prioritize swabbing pellet surfaces that have been in contact with other pellets, rather than in contact with the air or the ground where DNA may degrade faster. For very fresh piles, try to get as much mucus as possible on the swab. After you've collected the sample, insert the swab into the tube and use the lid to snap the swab, so that the tube closes with the tip inside. If the swab is broken too high on the stem, the lid will not close and you will lose the sample. Etch the relevant sample information onto the tube itself and record all necessary data in your field notebook, including sample number, sample condition, GPS coordinates, weather, and any other notes. If you will be resampling the same location, make sure to completely clear the pile before continuing to collect. A new sterile swab should be used for each sample. To avoid cross-contamination, you should not handle pellets with your hands or any tools that have come in contact with any other piles. Samples do not always make it to the lab right away, so if possible, store them in a cold place, such as a refrigerator after collecting for the day, to slow down DNA degradation. In the lab, the samples will go through a multi-step process to identify individual animals. First, DNA must be extracted from the samples. This occurs by putting the sample through multiple washes in buffer and some digestion incubation periods. Once DNA is extracted, it goes through the polymerase chain reaction or PCR process. Through PCR, our small amount of DNA for the genes of interest will become millions of copies, marked with fluorescent tags. These will be visualized on a sequencing machine to identify the genetic pattern of each sample and eventually match and compare individuals.